of medicine, sensing, and communication to share with us about their insights. Please type your question in the Q&A box in the Zoom window, and I will summarize the question in the end to the speaker. First, let me introduce Dr. Alec Zhang. He is Morton Sanger Professor in Oncology and Michael Rosenberg Scholar of Biomedical Research at UT Southwestern Medical Center. He learned his PhD degree in biochemistry at UIUC. After postdoctoral training at MIT, he established his lab at UT Southwestern Medical Center in 2007. He is studying immune surface molecular and developing novel therapy for cancer treatment. He published more than 100 papers and received many awards, including the prestigious American Society of Hematology Junior Faculty Scholar Award and Leukemia and Lymphoma Society Scholar Award. Now let's welcome Dr. Zhang. Dr. Zhang, could you share your screen?
Okay. Yeah, thank you, JC, for your nice introduction. Let me try to start to share my screen. Okay. So yeah, I would like to also thank the Chinese Institute of Engineers to give me the opportunity to briefly introduce the concepts of cancer immunotherapy. So let's get started with cancer. What is cancer? So according to Dr. Bob Weinberg, one of my former advisors at MIT, cancers have 10 hallmarks. So they can have indefinitive growth, and they are not easy to die. And then the growth depends on the blood supplies. So that is called angiogenesis. And then they can also migrate to distant tissues and organs, so namely metastasis. Importantly, the cancer can grow because they can escape the attack from the immune system. So our immune system is composed of a number of different cell types. So you may have heard about the name of T cells that can fight and kill the tumor cells, and also B cells that can produce the anti-tumor antibodies. So besides, there are a number of other immune cell types. They work together to fight cancer. So what exactly happened? So every day we have mutations in our DNA. And then this DNA mutation can may lead to the production of protein fragments, so called neoantigens. So that means new antigens. So these neoantigens only exist in the mutated cells, but not by the healthy normal cells. So our immune system has a way to detect the presence of these neoantigens by the mutated cells. In fact, the tumor cells can present these neoantigens on their surface by a molecule called MHC. And then another molecule on the T cells that called T cell receptor can recognize this MHC presented neoantigens. And then T cells get activated and then can kill these mutated cells. So that's the theory of the so-called immune surveillance. It was brought up much earlier. That's in 1957. And that's why, although we have mutations every day, it's not that easy for us to develop a tumor right away. Of course, over time, the tumor cells have a number of tricks to fool around the T cells and to suppress the T cell activity. So eventually, tumor can grow. So when the tumors grow up, they can even change that initially tumor-fighting immune cells and into the tumor supporting. And then this type of immune cells, together with other initially normal cells surrounding the tumor, form the so-called tumor microenvironment. So to effectively treat cancer, it's better to consider to attack both the cancer cells and the tumor microenvironment. So what's the option for the cancer treatment. So we all heard about chemotherapy. So that's widely used, but we also know it has huge toxicity. So then about 20 years ago, targeted therapy, which only attack the tumor cells, but try to spare the normal cells and tissues, became available. About 10 years ago, a new generation of cancer therapy called immunotherapy started to gain attention. So immunotherapy remains as the standard care for most types of cancer so far. So the chemo drugs can kill those fastly dividing or fastly growing cells because most of the tumor cells can grow fast. So the chemo drugs can kill them. However, chemo drugs can also to kill some of the normal healthy cells. So that's why chemotherapy always has side effects and toxicities. So besides, the chemo drugs can also spare some of the cancer cells because some cancer cells, such as some cancer stem cells, they don't divide fast. So they remain quiescent and cannot be effectively killed by these chemo drugs. So therefore, chemotherapy, although effective, but has high toxicity and cannot eliminate cancer. So in 2001, a magic bullet called Gleevec, which is the first cancer-specific tumor drugs get approved by FDA. So this strategy is called targeted therapy. So as the name implies, the targeted therapy is designed to directly attack the specific molecular lesion of the tumor, but not by the normal cells. So for example, Gleevec, which is the first approved targeted therapy as a drug, as I just mentioned, is an inhibitor of an enzyme called BCR-ABL. BCR-ABL only exists in the chronic myeloid leukemia cells, but not by any of the normal healthy cells. So that's why Gleevec is very effective to kill those leukemia cells. However, Gleevec, although effective, like any other targeted therapy drugs, eventually could lead to drug resistance. That's because the tumor cells are smart. So under the pressure of this targeted therapy treatment, then they could develop additional mutations. So that can get around of the attacks from the targeted therapy drugs. So eventually, inevitable resistance developed. So here is the cover of the Science magazine in December 2013. It marks the breakthrough of that year as cancer immunotherapy. So cancer immunotherapy, different from the chemotherapy or targeted therapy. It's a new generation of cancer treatment. So it's not only directly attack the tumor cells, but also indirectly attack tumor by activating the anti-tumor immune system. Because the immune system has long-term memory and also can attack multiple aspects of the cancer development. So this therapy is believed to be able to offer long-term disease-free survival or even cure of the cancer. So today I'll briefly introduce 3 types of new generations of cancer immunotherapy. So first about the checkpoint blockade, and then I'll talk about one new cell therapy called CAR T cells, and finally I will conclude with the personalized cancer vaccine. Cancer immunotherapy, as I mentioned, has long history. So in 1957, Thomas and Burnett, for the first time, brought about the idea about immune surveillance, as I just mentioned. In 1980s, the pharmaceutical companies start to produce one type of a molecule called interleukin-2, so which is tumor fighting or pathogen fighting molecule in the body. So they manufacture this molecule in the industry scale and then try to use it to treat different types of cancer. Eventually, it showed efficacy against a couple of cancers. However, it has very high toxicity. The reason is this type of treatment cannot have the tumor-specific immune activation. Instead, it induces widespread systematic whole body immune activation. So that accounts for its toxicity. So this type of result triggered some skepticism about the use of the immunotherapy for cancer treatment until about 10 years ago, in 2010, the first cancer vaccine developed by a company called Dendron became available. So Dendron basically isolated the immune cells from a group of prostate cancer patients and then treated them outside to enhance their immunity and then reinfused back into the patients, which showed enhanced anti-tumor efficacy for a number of prostate cancer patients. And then from 2011 to 2015, two antibody drugs called anti-CTLA-4 and anti-PD-1 revolutionized our concept about the use of cancer immunotherapy. So these antibodies, especially anti-PD-1, enable the tumor-specific immune activation instead to cause the systematic whole body immune induction. So this discovery won Nobel Prize in 2018, and these drugs, of course, get approved and showed good efficacy in treatment of a number of different cancer types. And then in 2017, a new cell therapy called CAR T cells was approved by FDA. So these three waves highlighted the peaks of the current version of cancer immunotherapy. And after that, of course, as always, we experienced some failures, and now we are regaining confidence for the whole direction. So now let me introduce these three strategies one by one. So first about Immu, checkpoint blockade. So this is still related to topics I mentioned before. So the tumor cells have mutations, and then mutations lead to the production of new antigens. And then new antigens can be picked up by the T cell receptor on the T cells. Then T cells get activated to kill the tumor cells. However, the tumor cells are smart. So when the T cells are approaching to try to kill them, then they can sense that, and then they can up-regulate one molecule on their surface called PD-L1. So this PD-L1 combined to another molecule called PD-1 on the T cells. Unfortunately, PD-1 is an inhibitor receptor. That means once it binds by PD-L1, then it can deactivate T cells. So suppress the activity of the T cells so that tumor cells get escape. So this mechanism is called immune checkpoint, namely the brake for the immune activation against the cancer. So after understanding this mechanism, scientists now can develop antibody drugs to block the interaction between PD-L1 and PD-1 so that T cells can be reactivated and then tumor cells can be killed. So as I mentioned, this discovery won Nobel Prize, and several scientists made a major contribution to it. So two scientists, James Allison, now is the chair of the Department of Immunology at the MD Anderson in Houston, and Dr. Hang Zhou, professor at Kyoto University, won Nobel Prize in 2018. And then another two scientists, Dr. Liping Chen, is a Chinese scientist at Yale University, and Gordon Freeman at the Dana-Farber Cancer Institute in Boston, also made important contributions. So as you can see, the antibody drugs showed very powerful effect for the cancer treatment. So the antibody actually is a Y-shaped molecule that is produced by the B cells. So it has the ability to specifically recognize and bind to another molecule called antigen. So any molecules can become antigen. So therefore, there are millions of different antibodies. So scientists now can produce antibodies in manufacture in large quantities. And now top 7 out of the top 10 best-selling drugs are antibody drugs. So it represents the future of the pharmaceutical industry. Of course, this 7, one of the top best-selling drugs include the anti-PD-1 antibody, as I just mentioned. So the anti-PD-1 therapy is highly successful. However, it's only effective in treatment of about 20 to 30% of the cancer. So why it cannot be effective in treating other type of cancers? So people find one correlation. They found the efficacy of the PD-1 therapy is correlated to a factor called tumor mutation burden. So tumor mutation burden means the mutation level in a particular type of cancer. As we mentioned, every tumor cells has mutations, and then this mutation can lead to the production of new antigens, and then new antigens can recruit T cells to attack these tumor cells. The higher the mutation level in the tumor, then the higher production of new antigens, and then the better ability to recruit T cells, of course. Then that make the PD-1 mechanism become more predominant. So then it's logical to see this correlation. On the other hand, this also suggests additional immune evasion mechanism that is independent of PD-L1 and PD-1 exist. So scientists need to make additional new discoveries to identify those kind of mechanisms in order to improve the efficacy of immunotherapies. Okay, then I talk about the second strategy, which is cell therapy, as I just mentioned, called CAR T cells. Somebody of you might heard about that name. So CAR T cells are the abbreviation of chimeric antigen receptor T cells. So this concept was brought up by Dr. Ashar. He's a scientist in Israel, and he brought up this idea in late 1980s. So this is something to do with the T cells and tumor cells, we just mentioned before. So, as we just talked about, the T cells has the T cell receptor that can recognize the new antigens presented by the tumor cells. So then Dr. Ashar proposed to re-engineer these T cell receptors by making two changes. One, she changed the external part of this T cell receptor to replace the native sequence with the antibody sequence that can recognize a specific tumor target. Because the binding between the antibody and the specific tumor target is much tighter than the native T cell receptor binding to neoantigens. So this makes the stimulation of the tumor cells to the T cells much stronger. Number two, he also changed the internal part of the T cell receptor. So upon stimulation, then that makes the T cells can proliferate much faster with much higher potency. So based on these two engineers, CAR T cells are produced, and in 2017, CAR T cells were approved by FDA for the treatment of several B-cell tumors. So because all these B-cell tumors have one specific tumor target called CD19. So as you can see, the efficacy of the CAR T cells really depend on the choice of the tumor markers. So if we don't have the ideal choice of the tumor marker, for example, if the tumor marker can also be expressed by some of the normal cells, then you would see toxicities. So this toxicity called on-target off-tumor toxicity. On-target means it can attack tumor. However, it can also have toxicity on those normal cells. So besides, CAR T cells also can have a cytokine storm, which is kind of side effects caused by the systematic inflammation response in the patients. So this sometimes can have a lethal effect. And in addition, CAR T cells can also enter your brain and cause neurotoxicity. Beyond that, the major problem of the use of the CAR T cells nowadays is that it can only be effectively used in treatment of blood cancers, such as leukemia, lymphoma, or multiple myeloma, which all come from the B-cell origin. And it cannot be effectively used to treat solid cancers like breast cancer, prostate cancer, ovarian cancer, pancreatic cancer. So what caused the problem? There are two obstacles. First, solid cancers like breast cancer or prostate cancer, they are more heterogeneous than the leukemia, which are blood cancers. So it's difficult to identify an ideal, unique tumor target on the surface for the design of the CAR T cells. Number two, as we mentioned before, solid cancers are protected by tumor microenvironment. So it's difficult for the CAR T cells to access and have activity against those solid cancers. So to expand the scope of CAR T cells, we still have a lot of work to do. The third strategy I'm going to talk about is called a personalized cancer vaccine. So as I mentioned before, in 2010, Dendron, the company, developed the first cancer vaccine. So that's a type of immune cells, basically called dendritic cells. So however, the use of that type of vaccine is time consuming because you need to isolate those tumor cells and the immune cells, and also to treat outside of the body and then reinfuse back into patients, and also expensive, as you can imagine. So in fact, Dendron, the company, got bankrupted after a few years, after this product was approved by FDA and showed efficacy because of the financial issues and the insurance company issues. So then scientists try to develop an easier use cancer vaccine. So again, this, the idea comes from the mutations in the tumor. As we mentioned again, the tumor cells produce mutations, and then mutations produce new antigens. New antigens then can recruit T cells and activate the whole immune system to attack the tumor. So and each individual cancer patient has different sets of mutations and thus different sets of new antigens. So by sequencing each individual cancer patients, we can learn the DNA sequences, and then by using a computer algorithm, scientists may be able to predict the individual sets of new antigens for each patient. And then based on that information, we can synthesize those new antigens outside the body and then inject the patients with these new antigens. So this idea actually proved to be successful in phase 1 clinical trials in treatment of melanoma and a couple of other cancers. However, it's still far from the real application. There are two problems. First, how accurate for the computer algorithm to predict the whole sets of new antigens for each individual cancer patient. So that's a real challenge. Number two, scientists probably need to understand more about the immune suppression mechanism in the cancer patients so that the injected new antigen would be more effective. So now you heard about 3 strategies of the current version of immunotherapy. You can imagine still there's a lot of challenges we need to overcome in order to improve the efficacies. So in the basic research, we need to understand more about the immune checkpoint mechanism. So for example, we need to study more additional inhibitory receptors besides PD-1. So PD-1 is just the one which led to the development of very successful therapy. And then if we study more of them, we may be able to develop more of the effective therapies. We also need to understand more of the tumor microenvironment, not only the tumor per se, because this offers the mechanism about how the immune cells interact with the tumor cells. And even more, even the germs like bacteria and fungi in your gut may impact the efficacy of the immunotherapy. So there's not more to learn. And we are making progress. So Dr. James Chen, a professor at UT Southwestern, also a National Academy member, also a board member of the Chinese Institute of Engineers here. So he discovered one new pathway called the Gas Sediment Pathway in several types of cells. So this includes tumor cells and some of the immune cells. So briefly, the DNA damage in those cells can activate this pathway, and then eventually can lead to the anti-tumor immunity. Then in some tumor types, this pathway becomes defective. So Dr. Chen is studying how to enhance this pathway in those type of tumor in order to develop novel immunotherapy. So Another example by my group, we are studying tumor microenvironment. So as we mentioned, there is an interplay between the tumor cells and the T cells. And the study of that interplay led to the discovery of the PDL1 and the PD1 mechanism, then not only won Nobel Prize, but also led to the revolution of the cancer immunotherapy. So besides T cells, other types of cells in the tumor microenvironment also can affect tumor development. So we identified one group of inhibitor receptors called LILRB. So these receptors expressed by other types of immune cells in the tumor microenvironment and enable the tumor microenvironment to suppress the T cell activity and to support tumor development. So by developing antibodies to block the activity of these inhibitor receptors, now we are trying to reactivate the T cells and suppress tumor cell development. So we have 2 clinical trials ongoing to test this idea. So what we learned so far? So yeah, as we talk about, immunotherapy is a new generation of cancer therapy. So because of the anti-tumor immunity has long-term memory and can attack multiple aspects of a tumor development. So this therapy may offer the possibility of long-term and disease-free survival, including cure of the cancer patients. So it's different from the previous chemotherapy and the targeted therapy. So what we need to do now is to identify additional new mechanisms in the basic research and also in the translational research about cancer immune invasion, so that we may be able to develop new immunotherapies which can improve the current PD-1 based therapy by combination. Also, another task we need to do is to further the development of CAR T therapy so that it can be used in treatment of solid cancers. And we also need to improve the development of personalized cancer vaccine, as I just mentioned. Still have a long way to go. And finally, we need to decrease associated with the immunotherapy. So in this way, we have to enable the tumor-specific immune activation instead of the systematic whole body immune induction. So finally, I would like to thank the trainees in my lab at UT Southwestern. It's their work made me possible to talk about this in front of you. And I would like to also acknowledge my collaborator, Dr. Zhichang An at UT Houston, who developed antibodies with us in the last 8 years, and Dr. Xiaoning Liao, a president of Immu company. So this company worked with us to further our clinical applications. Thank you, and I would like to take questions.
Okay, thank you very much. I like Dr. Zhang. So I would like to remind audience that you can type your question in the Q&A box in the Zoom. Then I will summarize them as Dr. Zhang. Before we got any question, I do have several questions myself. First, let me throw this out because we receive a lot of requests for speaker to talk about what do you see for the future in this field. There's a lot of concerned parents. Their children are in college or in high school. They would like to understand a little bit about in the research or industry field for cancer treatment or cancer immunotherapy. Can you shine some light on this?
Yes. So yeah. So overall, okay, I predict after 20 years, most of the cancer will become effectively treated and become chronic disease. So that's the overall perspective. So the reason is after we observe the hope from the development of PD-1 therapy, which is effective in treatment of about 20 to 30% of the cancer patients. So people are studying a lot of new mechanisms and trying to develop many new versions of immunotherapy. So I guess, within 20 years, a lot of this will become mature. So specifically, as I just mentioned, so in terms of basic research, we can, we should study more of the mechanism about how the cancer escape the immune surveillance and immune attack. So that's basic research. Of course, those basic research is highly related to the therapeutic development. So so it's very easy to translate into the drug development. So in terms of drug development, I think there are like 4 directions, as I just summarized. First, how to improve the PD-1 therapy. So the answer is, we need to combine the PD-1 therapy, which is already effective, with some other new therapies, which we may develop in the next decade. And also, how to develop CAR T cells so that it can be used in treatment of solid cancer. So still, this is also partially based on our better understanding of the mechanism of the immune evasion, because solid cancers have much more strategies or tricks, to get around of those kind of immune surveillance. So all this also depends on the, basic research. And also how to develop effective cancer vaccines. As I mentioned, one way is to develop better computer algorithms to predict the new antigens. So this needs some interdisciplinary collaborations. Number 2, also related to the basic research we are just talking about the basic mechanism of immune surveillance and immune escape. And finally, how to decrease the toxicity. So yeah, this one basically is related to how to enable to have the tumor-specific immune activation, so that we only want those kind of immune systems surrounding the tumor to get activated, but not activate the whole body's immune system. So of course, as you can imagine, this also related to some of the basic research. But I feel that the progress will be very fast, because so many outstanding scientists are working on that, and so much funding were put into it.
Yes, actually,